First of all, thanks to the library for having me do this talk. And my name is Cody Gilbertson, and I'm going to be talking to you about the endangered Chittenango Ovate amber snail. So we have a pretty large group of people that are working together on the recovery of this species. We work with two local zoos, the Rosamond Gifford Zoo in Syracuse and Seneca Park Zoo in Rochester. We also work closely with the New York State DEC and the United States Fish and Wildlife Service, the New York State Parks, and also SUNY College of Environmental Science and Forestry. So it's a really neat collaborative effort, which I'll be talking about. And I will start by giving you a background of why snails are important and what's going on currently in the world with snails. And then we'll move on to my graduate work, which is from 2015 to 2013 to 2016, excuse me. And then we'll talk about current updates that are going on with the project. So this is a picture of the endangered species biologist Robin Niver and myself in front of the Chittenango Falls, which is where the snails live. And to give perspective, I want you to look at this chart here. We have about 42% of all recorded extinctions are mollusks. So we have data on this coming in that show most of those, 99%, are non-marine, or so they're terrestrial land snails or slugs. So there's really a disproportionate amount of research going into mammals and other conservation efforts, whereas there's a pretty big need for conservation of mollusk species. So why are snails important? Well, we have a lot of diversity. They're second only to arthropods in the numbers of species that are out there. So we have incredible colors, shapes, sizes of different snails that are inhabiting both terrestrial and aquatic ecosystems. And they're, of course, a nutrient source for other animals. So we have birds can eat the snails and get calcium from their shells, and for their offspring provide calcium for the bird egg. And humans can also use snails as a source of protein. So these are giant African land snails that are worth eating because they're quite large and provide a source of protein for some humans. And the snails are also, in the nutrient cycle, they are kind of completing the bottom of the nutrient cycle. So they break down leaf litter and turn that back into soil that plants can use to grow. If there weren't snails inhabiting these niches, leaf litter would just sort of pile up, and there wouldn't be very good viable soil. So they're constantly working for us and for the plants and for the other animals. They're also known as an indicator species, which basically allows us to monitor the health of a forest. So in an area where there's a degraded habitat, you might find fewer snails in the area, whereas in a pristine habitat, you would find a lot of abundant different numbers of snail species. So in some examples of snails suffering in the wild, we have this glaring example of tree snails in two different families. We have Octanella and Partula. And these tree snails have been decimated by a predator snail that was brought to the islands. So they live in Hawaiian Islands and Micronesian Islands. And they did not co-evolve with predators, so they're naive to any kind of predator snail. And basically the predator snail will follow the trail of these guys and find them and then eat them. So 75% of all of the diverse species of tree snails have gone extinct because of habitat loss and this predator snail that was an introduced species. And one of the remedies to at least slow down the loss of this biodiversity is to hold a captive population of some of the remaining species. So at the University of Hawaii, they have this program where they've brought in the remaining snail species, and they breed them in the lab, and in a protected area. They actually have these exclosures for the snail that keep the predator snail from entering the wild area that they're released in. So some of these programs have been successful, again, in like slowing down the loss of biodiversity, but we don't know if it's going to be successful long term to maintain and preserve these species. Another example is the Partula tree snails, where at the Woodland Park Zoo in Seattle, they actually have a program where they're breeding the snails and they release them back into the wild in Tahiti, which they're tracking right now. So we don't have the data up to date on their survival, but they have released thousands of these tree snails back into the wild from captive efforts. So the snail that I work with is in this family, Succinea, and it's completely different than tree snails, but we kind of use this as a template to work off of as far as a captive effort goes. And our snail is temperate and living on the ground, whereas these snails are tropical and living in the trees. So it's a pretty different set of husbandry that's involved in this. And so the Chittenango Ovate amber snail, which I will call COAS from now on, it's a bit of a mouthful to say that the whole time. So COAS live in upstate New York, right in the center there where the star is, at Chittenango Falls State Park. And it's really kind of a unique species because it's not found anywhere else in the world. So this only area that it's found in is in our backyard. So COAS live here at this waterfall, and I'm a little bit biased because I obviously find these snails beautiful, and I think they're actually really cute. So they are tiny. They are about a thumb width in length, and they live for about 2.5 years, which means they overwinter upstate New York winters, and that's very impressive to me. Another interesting fact about them is their shell is actually transparent. So if you were to take the shell away from the body, they would actually be sort of just sort of clear or yellowish. And so that means that you can see their body through their shell, which is this beautiful marble pattern is actually their body. So that's kind of an interesting fact about them. And not only are they restricted to this one area at Chittenango Falls, their entire population lives on the left side of the falls over here. They're not even found up on the ledge up here. So their habitat and their entire population is about this in the size of a living room, essentially. They don't live on the right side of the falls or downstream, so they're not found anywhere else in the park, and they're not, as far as we know, there's no other population in the world. So they do have that as the #1 pressure to their population, because there's no other area that they live in. If something happened in that one specific area, then they could easily be wiped out. And the waterfall itself provides a bit of an issue because there's sometimes flooding and sometimes droughts. So during flooding, water can reach all the way over and wash away some of the vegetation of their tiny habitat. And during droughts, they can dry out. So then another kind of abiotic factor is the fact that they're nestled underneath these shale rocks. So there's this cove of shale way above them that on occasion comes down and crushes parts of their habitat. So you'll see later in the presentation some of the effects of the rock slides that occur. So not only that, there is an invasive snail species that has been brought over from Europe. We think that's called we call it species B. And pictured on my finger there is species B, the non-native snail. And you might have seen this snail in your backyard. We have it in our backyard. It's very widespread and it overlaps COAS habitat. So we did some analysis to make sure that there isn't so much of an issue with these guys unless there's a higher abundance. So the numbers add up that their density is actually not impacting their food resources, which is a good thing, but we make sure to remove these guys every time we're doing surveying. And there's also, of course, invasive plants. So pale swallowwort pushes out native vegetation that the snails might be using for refuse or consuming. So there's all these factors kind of piling up against the population, and that is why they are the most endangered species of New York State. They are federally listed as threatened, and the last estimation of their population that was published was in 2009, which is showing about 340 snails existing in the wild. In the past surveys, in the more recent past, in 2017, we are monitoring the population at even lower numbers. So we're getting 160. In 2019, it's even lower at 76. So these guys are kind of just really plummeting in their numbers in the wild. So how do we get that information? Well, we do mark recapture surveys, and that means that we actually have to tag the snails, which we put these little tiny honeybee tags on, and each one has a color and a number, and that way we can track actual individual snails and also run our data through an analysis and get an estimation of how many snails are actually out there based on how many snails we're recapturing. So the United States Fish and Wildlife Service has a recovery plan for COAS, and one of the goals on that recovery plan is to have at least 2 captive colonies so that we have a backup population to the wild population, as well as a way to potentially augment the wild population by releasing captive snails back into the wild. And in 1995, we had a grad student working on a similar project, and Andy Malloy, got some good biological information for caring for the snails. So this is a photo from 95, and the diet they were feeding was romaine lettuce and fish food. But ultimately, the last remaining snail in captivity passed away in 2002. So it wasn't a long-term solution. And there were some problems with it that there was a very high hatchling mortality. So within the first 2 weeks, 80% of the hatchlings in captivity would die. And there was also a pretty marked difference in the wild looking shell compared to the captive looking shell. So you see these ridges and cracks, and it pretty much, the appearance just doesn't look quite similar to what a wild snail would look like. So that was one of the issues that I wanted to address, is how to get the growth looking a little bit better. So as a grad student, I tried to tackle this issue and reestablish another captive colony. And my goal was to try to increase their survival, their growth, and also maybe we can get more reproduction in the captive setting too by changing some of the dietary stuff that was going on. So I wanted to look at what their food preferences were, but one of the problems was there's so few snails out in the wild that how do we work with those endangered snails without harming them while I'm trying to figure this out. So what did we do? We used a surrogate species, and we could use that that non-native snail that was overlapping with COAS habitat. Their scientific name is Succinea putris, but that we call them species B. So I used a bunch of species B. I looked at past research, and there wasn't that much information about people rearing snails in a lab. So there's just very little information about this, and I had to figure it out. Ultimately, what ended up working was observing one little COAS snail, and that was very effective, as you'll see. But working with the surrogate species, I tried lots and lots of different things. So here's a small amount of pictures that you see, leaf litter, vegetation from the habitat, which was nettle and watercress. I even tried the pertulid diet, which is something that they were feeding tree snails. And I used the romaine, I used carrots, and I even went out to the habitat to culture sticks and leaves to see if something would grow on an agar plate and see if the snails would actually consume something there. None of which that really worked. Here's a short list of other things that I tried. This really took a long time to get the diet sort of fine-tuned. And most of the people food just rotted. We have carrots, grapes, apples. Those things just weren't really touched by the snails, or they might nibble on it a little bit and then never come back to it. So once I tried the leaf litter, this is actually what was a game changer. So right away with species B, I noticed with the sugar maple that the snails were at least eating it. And then that's when I got my first reproduction of species B. So I knew I was sort of onto something with leaf litter. And I took the leaves that the snails pretty much like to consume, just a handful of sugar maple leaves or something that I found in the woods, and put some snails on leaf litter and some snails on romaine lettuce, just to see if there was any difference on their growth or survival. And sure enough, the snails on leaf litter grew faster and their mortality was lower. But for the romaine lettuce, there was actually higher reproduction. So we think that maybe the sugar content or water content in the romaine helped with something to do with the reproduction. So at this point, I thought I understood it enough that I could support the snail and get reproduction. So I tried to bring in 10 COAS snails from the wild, and basically we call it a snail year that happened in 2014. So unfortunately, when I brought those 10 snails in, they shut down, they didn't eat what I was offering, and their shells looked like this. They were dull in coloration. Their tentacles were kind of retracted, and they certainly were not going to reproduce under the circumstances that I was providing. So during that time, I got the most gray hairs of my grad school experience, and I had to end up releasing these snails back to the wild. But I was bringing all kinds of vegetation in from the wild to see if they would eat it, and I accidentally brought in a little stowaway snail that was 4 millimeters in size when it came in. So I didn't even know if it was COAS or species B. But I found out that it was COAS by this unique marking, a little black spot on their foot, and asked the DEC for permission to see if I couldn't observe this one snail and get it to live and eat something under very close observation. And that's when I like to say we snailed it, because from this one little snail, we got really good information on what the feeding preferences are. It was easier to tease apart because I could chart its growth corresponding with the different foods that I was feeding. So over here at E, I started feeding cherry leaves, and we see the growth kind of like take off exponentially over time. It tapers off because they only grow to be about 21 millimeters. So these are cherry leaves. I collected the leaves from the same tree at the same time, and there's a bunch of leaves that they don't touch and some that they do. So the mystery continues as far as what they like to eat. And we found out that these translucent leaves that you can see light through, so the thinner leaves, are really highly preferred, and you can see the feeding holes here and feces. So we know that they were feeding on this leaf. Whereas the leaves that are thicker and more, I guess, you can't see light through them. That's how we gauge if they're thick or thin. The snails don't touch those. So that's one main characteristics that we can describe about cherry that they prefer. Again, this is a fire cherry leaf that they've consumed. So there's feeding holes there, and snail poop, which tells us a lot. So with sugar maple, it's the same story. We kind of just collected at the same time, and there seem to be these really specific characteristics that the snails preferred over others. So these darker pigmented sugar maple leaves, the snails don't touch, and the sun-bleached sugar maple leaves, they really like. So all of this was just found out by putting leaves in with this tiny snail and seeing what it would like to nibble on and going out and getting more. So the new diet is basically that. We have a mixture of sun-bleached sugar maple leaves, and then we have cherry leaves that were thin and light. You could see light through them. We also put in some romaine lettuce, and this white stuff is calcium supplement. So with that, this snail on the bottom is our little stowaway snail that grew really monstrously large. And you can see that this is during cherry feeding. You can see that the snail has actually grown very smoothly, and the body is very vibrant. So it's kind of neat to see how well it was growing off of these leaves. And I conservatively tried bringing in 2 more adults from the falls. That way we could see if it would actually translate to the wild adults as well. And it did. And basically it was love at first sight. We had our first reproduction and mating occur virtually upon placing the snails together. And so they're hermaphroditic, which is an interesting fact about them. They both fertilize each other. And then that means that they can both lay egg masses. So that actually is to our benefit. We have a bit more numbers of eggs in the lab because of that. So 7 days after mating, they laid their egg masses, and it was a really interesting process to watch and very exciting. We had lots of egg masses laid in interesting places, like the base of this stick. And when you kind of take them out and examine them, they kind of look like golden grapes. And but they're actually, when you pull them apart, they're actually on one continuous string. So they kind of look like Mardi Gras beads, which is really fascinating. I don't know why it's like this, but this is an up close microscopic look at the eggs. So we tracked the embryonic development of them because that has never been done before. And within about a week, they had a little snail shell kind of developing within the egg. Takes up about a third of the egg, and then within a month you see these little eye spots develop. So we were seeing these little babies develop, and their eyes are basically just staring back at us. And this was an indicator that they were about to hatch out, so I would put leaf litter in at this stage for them. And when they hatch out, they look like this, which I think are one of the most adorable things ever on the planet. So they're only about 1 millimeter in size when they first hatch out. And you can see this is actually a leaf vein. So this is one of those cherry leaves, and that's the vein in the leaf. And so that's how tiny they are. They are translucent yellow when they first hatch out, and then within a couple of days, they darken up a little bit. So this, I would say, is a toddler, maybe like five days old or so. And the coloration of the shell is a bit darker. And then fast forward 8 months, and we have these guys. They all turned into beautiful adult COAS snails that their shells are vibrant, their growth is looking really good. There's poop everywhere, which is a great sign. So this is again calcium, and their tentacles are out, so they're active. All of these are good behavioral, physical signs that they're doing pretty good. So for a quantitative look at things, we had within the first 3 months even 0% hatchling mortality, virtually zero. Whereas again, in the past, there was over 2 weeks, there was 80% of mortality of the hatchlings. So the leaf litter diet was far superior in supporting their growth and survival. And even over 2 months, there was 19 egg masses and 632 snails. So we had an explosion of reproduction just from 3 snails. And in the past, they had 6 snails over a year, an entire year, lay 130 eggs. So, and you know, most of those didn't survive. So we had pretty stark differences with the change in diet. We can also see a side-by-side of the comparison of the shells in the past. These are the 1995 photos, and 2015 photos of the snails with the leaf litter diet. So again, quantitatively showing that the snails like these transparent cherry leaves, we did a weight study. So we weighed the leaves before and after the snails fed on them, and then measured how much they were actually eating of each one. And we see that the cherry leaves that you can't see light through, the really thick ones, were not preferred really at all. So again, we have some of these characteristics of leaves that they really do highly prefer identified. And there was a pretty major difference in the husbandry, survival, reproduction, and growth. And then leaf litter was often not fed in the past research that I was looking at. So I think that for future conservation efforts, leaf litter needs to be examined for any kind of supplemental feeding for an endangered snail. And everything's not perfect. So we're still working on lots of issues going on in the lab. That one is that we're actually really inhibited by the food supply. So we can't maintain 50,000 snails. It's just not possible. There's not enough time in the day. There's not enough leaves. So we are limited by our food supply because we're hand collecting these leaves. And if we don't have enough to support the babies, we don't want to rear them. So we have many eggs that we rear, and then some we preserve, and hopefully we can use the eggs in the future for any kind of releases. And there's also nematodes. So because we're looking at a holistic diet and we're collecting these things from the wild, we're also bringing in nematodes to the captive population. But because we change the terrariums every week, we limit the overall abundance of the nematodes so they're not harming the snails. And that hasn't been any bit of an issue in moving forward. But you know, we constantly kind of worry about what if we bring in something that is going to be harmful for the snail. And so we process the leaves, we dry them, and we store them dry for 2 weeks at least before we put them in containers to use. So we're trying to take precautions, but you never know if something accidentally could get in there. So 2 backup populations have been established. We had a colony of snails at ESF, and we had a colony of snails at the Rosamond Gifford Zoo. And each one has about 500 snails. So if we're looking at the total population numbers, we have well over the number, the actual wild numbers in the wild population. Again, there's the last estimate was around 76 in the wild. So we're supporting 5 times as many snails in the lab, and we've had over 50,000 eggs, or at this point, there's probably 100,000 eggs documented to have been laid in captivity. So obviously there's no way we can support that many, but we are doing our best with supporting the adult snails as they come. So another interesting thing that happened from the grad work was the ability to be able to release these snails back to the wild. And we had beautiful, healthy adults that were tagged that we released it in 2017, 20 adult tagged snails. And then also in 2015, we had that explosion of babies. So we did release 270 of the little baby snails back to the habitat, and here's us being very excited about that. The first ever release that was able to be done. And at this point, I guess like this is sort of the end of what I accomplished as my grad work. So from here on, I will discuss sort of the future aspects of the project and other aspects that are going on. We did do our first survey of the wild population this year in June. And last year we did not do a survey due to COVID, and also the numbers decreased so much that we wanted to give the habitat a break of people getting in there and trampling around. So this is a very, very exciting finding from just a week ago. This is actually one of our captive reared and released snails that we found back in the wild. So we released them in September, and that means this particular snail grew up in the lab, went out into the wild, overwintered, and now was found surviving. And this is one of 3 that we found out there last survey. So we're really excited that at least we know that they can make that transition from captive life to going back into the wild. We do have a new release plan, and rather than releasing only adults or only juveniles, we have decided to release all 3. So we're actually putting some of the egg masses, juveniles and adults out this year, and hopefully kind of just making an even age distribution. We're going to put more eggs out and fewer juveniles, and then even fewer adults to sort of see if that balances out their life. What do I want to say? If it's an even balance of the sort of pyramid out there in the wild. And we also bring in new founders occasionally to hopefully increase the genetic diversity in the captive population. So this year we also found 4 new snails that were not marked, that were not captive release snails, and brought them into the lab to integrate into the captive population. What we do is we bring them in, hopefully they mate and lay egg masses, and we take the egg masses and then we release the adults back out to the falls. So we're not actually taking adults away. So another aspect of the project is sort of habitat management, and I wanted to show you in 1995, this is a photo from the habitat, and I'll show you in 2015, we also have a photo of the same area. And COAS live kind of in through here, where we see this is early spring, so there's not much vegetation growing yet, but you can see that this is a fairly large area that would be greened up over time once spring progresses. And in 2015, we have that same area with the 2006 rock slide, which was a huge boulder came down. And you can see this is basically the same area that all these rocks are covering. So again, a little comparison. And so this has been quite detrimental to their population, we think. And the New York State Parks is looking at potentially hand chipping up some of these smaller boulders and removing those out of the area, trying to get some of this stuff cleared up, and replanting. Hopefully we can replant some of the native vegetation up here, so we could bring in some soil or just replant some Joe Pye weed, which is a plant that they really like. It's abundantly found over here. So those are some of the ideas that we have to actually manage the habitat. And also we do remove pale swallowwort, which is that invasive plant that's in the area. That's a very tedious thing. We've been staying on top of removal, and we've actually seen an increase in nettle plants that are growing in the area that we've removed pale swallowwort in. So it's somewhat effective, but it's just a nasty plant that's just impossible to get rid of. So those are some of the things that we were thinking about, but also one of the ideas is to plant a cherry tree or a sugar maple tree or something to have more leaf litter fall into the habitat. But that hasn't happened quite yet, and there's a lot of trees up here that they do feed on. Over time, those leaves decompose, and it's primarily red oak that's in the habitat. But we have found the snails on those leaves. So another aspect is basically searching for another habitat area that COAS could inhabit. And we have a postdoc, his name is David Bullis. He's working on just that. So he is measuring temperature, humidity, vegetation. He's looking at different area waterfalls to see if the landscape might be able to support a population of COAS. And we could use our captive snails for a release into a different area, or also release some of those extra egg masses that we get. So ongoing things are occurring with the leaf litter. We are basically continuing to try to fine-tune our understanding of exactly how to process the leaves and collect the leaves and be able to have a good store of those leaves to support enough snails in captivity. But I want to say that this setup I thought would work because we have rain and sunlight that can decompose the leaves, but they're kind of held down in one spot so the wind doesn't blow them away. And the thing is that this doesn't work because, again, the different thicknesses of the leaves make it so maybe 10% of these would be decomposed enough for the snail to eat, but the rest of them would be not ready yet. So it's just a hodgepodge of of a headache. And we did find that hand collecting is the best method, but it's just very tedious. So we hand collect these sun bleached sugar maple leaves. We did find that they like pignut hickory, and most of these are decomposed enough to rake up and just basically sort out the oak leaves that come with them. So this is somewhat effective in time. We can get maybe 6 bins of these ready in a few hours by sorting through the oak. They do like green ash and also pawpaw. So we're finding out that they like some different types of leaves, and we just have to stay on top of at what time do we collect them and what characteristics do these picky snails like. So from now on, I will talk about our outreach efforts and kind of the public awareness part of things. And anybody who's watching, I would like to invite you to like our Facebook page, and our collaboration is called the Snail Blazers. So you can get up-to-date posts and funny things like this, whereas we're celebrating a birthday party for this one snail. But if anybody's interested, just type in Snail Blazers on Facebook, and you can get these up-to-date information on the project. We had the birthday photo shoot on document here with the macro lens and our little birthday background. You can see really these tiny snails have their birthday hats on, but that's how tiny the snail is in actuality. And then imagine their babies, they're even smaller. So this is a photo that came from that birthday shoot. This snail is very special to me because the little hatchling snail that came in as a stowaway had offspring, and this is basically, I call this snail Hatch Junior. And this snail lived to be 5 years in captivity, which is twice the lifespan in the wild, and it's the longest recorded lived snail in captivity. So I'm particularly attached to this snail, and I was very sad that it passed away, but very impressed that it lived to be 5 years. And these tiny birthday hats were a pain to keep them on their shells. They kept falling off, so I had tweezers. We also do snail racing, and this is a really good board for snails because you want to put them in the middle. We do not use COAS, we use the species B snail. So anywhere you put them, any direction that they go, excuse me, they will cross a line eventually. So kids really like to bet on who's going to win. They like to name the snails, and it's a really good interactive thing that even adults like. So this is our assistant Ashton Yost performing a snail race, and we have zoo camp kids gathered around. They're very engaged, and I think it's a really good activity. Even the 9th graders liked it too. So that's been a fun sort of addition of what we do for outreach. And then we have a kids booklet that we distribute at the Rosamond Gifford Zoo, or tabling events that we do. I'll see if this works, the link here. So we also distribute this at local libraries. And so this is the little children's booklet, and I'm going to zip through this, but really colorful photos, nice, engaging, cute story about a snail called Chit. We do call them the Chit. And pictures of the waterfall. This is my favorite photo of baby snails. The rock slides, and beautiful photos of our captive snails. And then our
Cody, we're not seeing the book isn't coming live on our side.
Oh. Well, new share. Let's see if this works.
Yes.
Can you see it now?
Yes.
Okay. Let me start over here. Well, I'll just go backwards. Beautiful photos of the waterfall and cute photos of the snail. And it's just a really cool interactive book. And our group there, birthday celebrations, and different leaf litter that the snails like to eat. So let me see if I can't get back to the regular page. Okay, did that work? Can you see?
Yep, you're good.
Okay, excellent. All right. We also have a snail sculpture that was built for the Rosamond Gifford Zoo play area, and kids like to play on this. It's right past the outdoor birds. It's a great way to engage the public, and I don't know, they did a really excellent job with anatomically covering how the COAS look. And here's some kids playing on the snail sculpture. And of course, for our December meeting, we had to put a Santa Claus hat on it, or Snail Claus. And we also teamed up with a local brewery, Critz Farms, where we have a snail ale as a fundraiser for awareness of the snail. And also all the funds went directly to the zoo for the conservation of COAS. And we also have these t-shirts. I don't have any for sale or any, we ran out of them, but we have the Who Gives a Chit shirts. And on the back, they say, I do, with a picture of the snail. So we have hopefully sparked interesting conversations by that t-shirt. And if anybody is interested in donating to the project, there's a donate button through the Rosamond Gifford Zoo website. So if you just go to Rosamond Gifford Zoo, right on the front page there is a donate button, and the only further instruction would be to, in the comments section, write that it's specifically for the Chittenango snail work. Otherwise, it just goes to the zoo, which is also a good cause. But if you want it to go directly to the snail project, you have to specify that. So with that, I would like to say we need to look back at the big picture here for snails in general, and with the knowledge that basically this is just one species that we're working so closely with, but any conservation information, new knowledge that we get from this work hopefully can be applied to other conservation efforts in a wider global effort. And we're finding out new information all the time. We have to understand what works and what doesn't work, and hopefully get that information out there and apply that to other conservation efforts. So I am pretty much finished with my presentation, but I really like to talk with people, so I can use the rest of the time to engage with questions. And I would like to thank the Fish and Wildlife Service, Great Lakes Restoration Initiative for the funding, and all of the collaborators. This has been a really interesting and challenging project to work on, and the snails definitely have my heart. So with that, I can answer any questions that people have.
Okay. It was a really great talk, and I had no idea. Thank you.
Yeah, there's a lot to the project. Yeah.
They are really cute.
Yeah, they definitely have stolen my heart. I love these guys.
Okay, then I'm not seeing any questions.
Okay.
Okay. Thanks, everyone.
Well, thank you for listening. I don't know if there's if you want me to wait around for a little bit, or